All right, biology students. During our last face-to-face class, we went over exploration 1 and went over the basic concepts of cellular respiration. And today we're going to focus in on more of the details, shall we say. But as we're focusing on the details, I want you to really think about this diagram here and how photosynthesis and cellular respiration are very, very similar processes in a lot of ways. Besides, as we talked about, the reactants for photosynthesis are the products of cellular respiration, and the products of photosynthesis are the reactants in cellular respiration. And so, this light energy and water that's being used in photosynthesis is really making all of these chemicals needed for all living organisms to live, essentially. Okay, and the main byproduct between all these things that the organism is using is the ATP, and the excess amount of waste product, all right, is given off in the form of heat. Okay, so those are the main things. So these two things are working together as a cycle and are dependent upon each other. Oops, I thought I paused it, but apparently I didn't. Okay, so moving on to exploration 2. So using this chemical energy. So remember, the name of the game is homeostasis. You got to maintain it in order to provide the cells with enough energy to do their daily functions. So bonds of food molecules are broken down with the help of oxygen, and they are forming other helper molecules like ATP, and in order to drive the whole process. All right, so moving down here, this is a really important diagram here. Now, when glucose comes into the cell here, right? When glucose comes into the cell, it goes across the cellular membrane. It's out in the cytoplasm, right? And in the cytoplasm, the first part of cellular respiration takes place, which is called glycolysis. Now, glycolysis basically is, for all organisms, takes place in the cytoplasm of the cell. And glycolysis breaks down this glucose molecule, basically splitting it in half. When it does that, it releases ATP in the cytoplasm of the cell. Now, from that part, those glucose molecules, after it's broken down, is going to enter what's called the form of what we call pyruvate. And pyruvate is the entrance molecule into the Krebs cycle, which we're going to discuss today. All right. And we're going to learn why it is a cycle. And this is releasing a lot of ATP. All right. And so when glycolysis is broken down, you have some electrons that are carried in the form of this other currency molecule, NADH, over to the other part of the respiration process called electron transport chain. So the oxygen is coming in, okay, and it's picking up these hydrogen electrons to form ATP, and water is a byproduct. Okay, so the Krebs cycle products are carbon dioxide, and the electron transport chains are water. Okay, so just like photosynthesis, it's a two-step process. Remember, in photosynthesis, the light reactants and the dark reactants, and one input was going to one and the other input was going to the other. Very similar. Maybe that question will show up on your test. Maybe. All right, so we'll go over this today in class. Glycolysis, like I said, when glucose is a 6-carbon chain, all right, when it comes into the cell, ATP, all right, is helping break that apart into two what we call pyruvate molecules. All right. And so this ATP is using the energy and it's releasing an ADP. All right. And then ADP is coming over here and picking up another phosphate group to go to ATP. All right. And becoming energized. So it's like the recharging of the battery. Same thing with NADH. Right. It's picking up some electrons there and hydrogen molecules from the water, and bada bing, bada boom. Next thing you know, you got two pyruvates, okay? And so it's really important to know that this process is releasing ATP, right? So you got to use energy to get energy, right? So that's how life works. So let's move down here to the Krebs cycle. The Krebs cycle, it's another one of these things that was discovered and won somebody a Nobel Prize. It's also called the citric acid cycle, right? Citric acid cycle. You guys know what citric acid is, right? Also known as vitamin C, right? I think that's what it is. It's the first step, first set of reactions in cellular respiration. And the name of it is discovered by Hans Krebs. Man, that sounds like a German name. The cycle is a continual series of reactions. That's why it's called a cycle, because it's cyclical and it never basically stops. Okay, so the function of the Krebs cycle is to complete the breakdown of glucose started in glycolysis. All right, so take that pyruvate that is entering the mitochondria and undergo a series of reactions to get some energy out of it, shall we say. All right, so the name of the game is produce this ATP. All right, so this is done by transferring high energy electrons to the electron transport chain, which is the second part. Okay, so when you are looking at your digital book or looking at your paper book, all right, we'll go here and start at number 1.
1. Pyruvate is broken down. 2. A 3-carbon pyruvate molecule is split into a 2-carbon molecule and a carbon dioxide molecule, which is given off as waste. High-energy electrons are transferred to NAD+, forming a molecule of NADH. The NADH moves to the second stage of cellular respiration, the electron transport chain.
All right, so that 3-carbon pyruvate enters into the mitochondria, all right? NADH comes over here and move some of these electrons. One of these carbons is released in the form of carbon dioxide, right? So think about it. The O2 here and the H here, that's coming from water. All right, so you have to have water in order to run the Krebs cycle, right? That's just really important. If you become dehydrated or for some reason you don't have enough water in your body, you cannot run the Krebs cycle very well, okay? So one carbon dioxide is released here in the form of a waste product, okay? And so now you go from a 3-carbon down to a 2. All right, so let's see what happens in step 2.
2. Coenzyme A is added. A molecule called coenzyme A bonds to the 2-carbon molecule, forming an intermediate molecule.
All right, so this coenzyme A is an enzyme that attaches to the 2-carbon, and when it enters the cycle here, it's going to help in the formation of what's called citric acid, which is a 1, 2, 3, 4, 5, 6 carbon atom, right? So here's 2, right? And it's going to bond over here with these 4 from here to form a 6-carbon citric acid.
Citric acid is formed. 4. The 2-carbon part of the intermediate molecule is added to a 4-carbon molecule to form the 6-carbon molecule called citric acid.
All right. So citric acid is then under NADPH coming over here, grabbing some more electrons. And when that happens, carbon dioxide is released. So it goes down from a 6-carbon to a 5-carbon.
Citric acid is broken down. 5. The citric acid molecule is broken down by an enzyme, and a 5-carbon molecule is formed. A molecule of NADH is made, which moves out of the Krebs cycle. A molecule of carbon dioxide is given off as a waste product.
All right, moving on to step 5. You got your 5-carbon here, and now it's going to go down to 4-carbon through a couple of processes.
5. The 5-carbon molecule is broken down. The 5-carbon molecule is broken down by an enzyme. A 4-carbon molecule, a molecule of NADH, and 1 ATP are formed. Carbon dioxide is given off as a waste product.
Okay, so carbon dioxide is given off as a waste product. All right, this NAD plus comes in here and recharges back to NADH. Okay, this ADP coming in here and recharging to form ATP. And now we're back over here to the last stage of the Krebs cycle, which is step 6.
The 4-carbon molecule is rearranged. Enzymes rearrange the 4-carbon molecule, releasing high-energy electrons. Molecules of NADH and FADH2, another electron carrier, are made. They leave the Krebs cycle, and the 4-carbon molecule remains.
Okay, so it's really important to point out here, this 4-carbon molecule here is now going to be attaching to coenzyme A with this 2-carbon. Sorry, I had to sneeze. This 2-carbon molecule here, and the cycle continues. Okay, so this is real important to understand that this cycle is never breaks down carbon to just a single individual one. It never goes below the 4-carbon chain. Okay, so it's just a matter of recycling and reusing these molecules through these enzymes rearranging their functions. And doing so, they either release energy or pick up a different amount of electrons. Okay, so we'll go over this one in class today. I'm going to call on some people to fill out this one. Same for here. Moving right along. So the main idea here is the Krebs cycle is a continuous series of reactions. All the carbon atoms in glucose eventually end up as carbon dioxide as a waste product, which is then kicked out of the cell. The role of electron carriers NADH and FADH2 is to transfer electrons to the electron transport chain next to the next stage, which is the next stage of respiration. These transfer electrons will fuel the formation of ATP. Okay, so moving on here to the electron transport chain. All right, so the second part is the electron transport chain. The second stage, which is the second stage of cellular respiration. And it uses these proteins embedded in the intermembrane of the mitochondrium. It's similar to the electron transport chain stages of photosynthesis. How interesting. The energy carried by NADH and FADH2 molecule produced in the Krebs cycle is used to make ATP here in this second step here. A number of enzymes are involved in this process. All right, so what we got here, we got the intermembrane, and you got some proteins that are embedded into the phospholipid here, right? And you got what was called the matrix of the mitochondria. This is the inside part, right? So essentially what happens is electrons are being transferred along this little protein channels across here. Sometimes you refer to it as the bucket brigade, where one thing is passing, an electron is being passed down here, right? I think here's a good analogy here. When people hear passing the sandbags along the in the form of a line, so people were just passing these along, bada bing, bada boom. That's essentially what's happened here. These high energy molecules, or NADH and FADH2, are picking up are transferring or carrying these electrons over here to this process and releasing them. And when an NADH is released, right, all right, it releases a water molecule here, right, and that is then pumped across the membrane here, right? And so same thing here over here when the FADH comes over here and picks up, has its charge, shall we say, and then it releases the electron here, right? And when it releases the electron, it's able to pump one of these hydrogen molecules off. Now take a look at the charge of an electron and take the look at the charge of the hydrogen. How do you think it's moving across this membrane? I wonder. So moving right along. So we get down here to the end, and these electrons are continuing to pump hydrogen across the cellular membrane here in the electron transport chain. Okay, and once it does that, the end byproduct over here is hydrogen is going to, some of the leftover hydrogen is going to combine with oxygen molecules and form water, and the water is the byproduct of the electron transport chain. Okay, so the last part down here is ATP is produced. Just like photosynthesis, the flow of electron ions is used to make ATP. All right, hydrogen ions diffuse through the protein channel in the inner membrane of the mitochondria. This channel is part of what we call the ATP synthesis. In other words, making ATP. ATP synthesis adds a phosphate group to ADP to make the ADP molecule. All right, and so that's the name of the game right here, is to use these high energy molecules to form the ATP, which is ultimately what we're using in the form of energy. Last but not least, like I said before, oxygen picks up the electrons and hydrogen ions to form water. So water is the end result of the waste product here. Okay, so we'll do this one in class here, and do this one in class. Here's the main idea here. Together, do glycolysis and respiration produce a net gain of up to 38 ATP molecules for every glucose. Okay, cells use ATP to carry out activities, right, such as active transport of molecules and ions that help maintain homeostasis. Okay, if somebody has the big brain today, you can tell me how many ATP are produced in glycolysis and how many ATP are produced during the electron transport chain and how many are produced during the Krebs cycle. All right, so they all have to add up to approximately up to 38. So somebody else tell me today, what are some factors that could limit this process not to be able to gain up to 38? What are some factors that you think could possibly prohibit it from being this efficient? Let's say what if it only produced 28 ATP? What do you think? Who's got the big brain today? Okay, so cells, the last part in fermentation, we talked about this the other day, so I'm not going to spend too much time on it, but the main idea is you should read it for the specifics. But nonetheless, if you run out of oxygen, right, let's say you're exercising, you're trying to train for the triathlon, for instance, right, you're going to be doing lots of physical activity. Right, this process, you can't get enough oxygen into your cells in order to break down this process efficiently, right? So you don't make enough ATP and you don't have enough oxygen. So what happens? The cells basically cannot do full respiration. So they can, however, produce ATP without oxygen, and through the process of anaerobic glycolysis and fermentation, right? So fermentation, right, does not make ATP, but it allows glycolysis to continue. Maybe that's a test question. Fermentation moves electrons through NAD+, H molecules and recycles NAD plus molecules for glycolysis. So basically, it's continuing the process somewhat, but it's not as efficient. Okay, so this is important because glycolysis, like respiration, needs molecules to pick up electrons, and it needs molecules of NADH. So the role of fermentation is to simply provide glycolysis with a steady supply of NAD plus. When you feel your muscles burn during a hard exercise, that is the result of fermentation. You can actually feel it. So lactic acid is the waste product of fermentation that builds up in your muscles, which causes that burning feeling. And the next day, that is why you feel very, very sore. And so lactic acid is quickly broken down. Once oxygen is available again, your cells can return to using cellular respiration. So this is why it's extremely important. After a hard days of exercise, you should actually go out and get your heart rate up in order to get as much oxygen in your body so you can finish breaking down lactic acid and complete the cellular respiration process. So you just got to stop being lazy and go out there and do it. Okay, so we talked about this too, the fermentation of different microorganisms producing, some of our favorite food products. All right, so that's pretty much it for today. You have 2 assignments that you need to do. Please check your Schoology page. Those are going to be due on Monday. Make that magic happen. I think we're looking at having a quiz on Monday or Tuesday, so please get this done ASAP. Okay. All right, we'll go over this a little bit more in class today. Ciao.